Good afternoon and welcome to Cure Talks. I'm Priya Menon, your host, and today we are discussing the new FDA-approved gene therapy for reversing blindness. Our guests are none other than the doctor couple responsible for this breakthrough approval. Dr. Jean Bennett, F.M. Kirby Professor of Ophthalmology at the Perelman School of Medicine at the University of Pennsylvania, and Dr. Albert M. Maguire, Clinical Associate of the Division of Pediatric Ophthalmology at Children's Hospital of Philadelphia and Associate Professor of Ophthalmology at the University of Pennsylvania. Welcome to Cure Talks, Dr. Bennett and Dr. Maguire.
Thank you very much. Happy to be here.
On our patient panel, we have Tabitha Mitchell, mom to Emery, an 11-year little blind girl. Tabby and Amy have been changing what it means to be blind since Amy's diagnosis with LCA at NIH when she was 3 years old. They tirelessly advocate for rare disease research and blind children. We also have with us Sarah Pettit, mom to 9-year-old Creed Pettit, who has received the newly approved treatment Luxturna. So once again, a very warm welcome to the guest and the audience. We will be addressing questions from the audience towards the end of the discussion. So if you have a question for the panel, please email your questions to priya@trilix.com, or you can also post your questions in the comment sections on curetalk.com. So beginning with the discussion, Dr. Bennett, you bred a virus to carry the gene, and Dr. Maguire, you injected it into a patient's eye, and there was light. So this is an incredible journey. I would like to start with a very, very basic question, Dr. Bennett. What is a person who has Leber's congenital amaurosis actually be able to see? And what causes this condition?
Sure. I'm going to start with a general answer, and then Al Maguire can be more specific. So there are at least 18 different genetic flavors of Leber's congenital amaurosis. Those are caused by different mutations in different genes. And depending upon the exact genetic cause, people have different levels of light perception versus vision early in life. And most of these genes actually cause damage or cause malfunction of the photoreceptors, and that explains the visual findings. So for the form that was just where we've been developing the gene therapy that just received FDA approval, that's a form caused by mutations in a gene called RPE65. And I'll pass it over to Al to describe what people can see with this condition.
Well, the RPE65 helps load nerve cells with the molecule that captures light called 11-cis retinal, and that's not functioning correctly in people with this particular form of Leber's amaurosis. They have some vision, and in some cases it's enough to get them, they're able to function and get around a bit when it's very, very bright. The way I think of it is they have about 1/10,000th to 1/50,000th the sensitivity to light that somebody with a normal retina would have. So it's as if you had a rheostat and you kept turning it down so that they are only able to use about 1/10,000th the light that you or I, and you can imagine that would make things difficult, especially when the at hours of the day when it's not as bright out. It's sort of like a enforced curfew. There's also an issue with the central vision where there's some blurring because the resolution, the sharpness of vision in the center is reduced as well. So overall, it's dimmer and blurrier.
Mm-hmm. So Luxturna is approved for people who carry the RPE65 mutation. Essentially, we have a gene therapy which replaces this. So can you talk a little bit about what we will be exactly doing in the gene therapy here?
So, as you said, people who have this particular form of LCA would be candidates, and what is done, which is very similar to the protocol followed in the phase 3 trial, the efficacy trial, is we establish whether people have enough functioning cells that they're going to get some useful vision as a result of the treatment. If you have no cells, because over time they've wasted away, it really doesn't make sense to do the treatment. It's not for people who don't have functioning retina. So once it's established they have potential and enough cells, they undergo a procedure called vitrectomy, where the back of the eye behind the pupil is entered with tiny instruments, and the Luxturna, which is the gene therapy drug, is injected under the retina in the vicinity of the nerve cells, and those nerve cells will take up the drug. So that's done in one eye, and after anywhere from after 1 week, the other eye is treated as well. And while all this is being done with the surgery, the patient has a course of corticosteroids, something analogous to the dose you'd use for asthma or poison ivy, and that's to tamp down any possible immune response to the drug, since it is a modified virus. Fortunately, we really never have seen much, if any, response to the drug in terms of inflammation.
Mm-hmm.
And so just to sum up, yeah, sure. Go ahead.
Yes, Dr. McGuire just mentioned the virus. So Luxturna is also the first AAV viral vector product approved for clinical use. So Dr. Bennett, it would be really good if you could talk a little bit and explain what an AAV viral vector is for our audience.
Sure, and as you said, this is the first first viral vector that's approved for direct delivery for genetic disease in the United States. And we're very excited about this. And so what this is essentially a shell. The virus typically has a shell of proteins which package the genetic material, in this case DNA. And viruses do what they do very well. They bind to cells and get into the cells, and then the DNA, or for other viruses, RNA, makes its way to the nucleus, where it takes up shop and starts encoding for the proteins that are encoded by that particular DNA strand. And so this recombinant virus is designed such that the bulk of the viral DNA that for the wild type virus, which can be copied in the cell, is removed, and instead it's replaced with the gene of interest, in this case of RNA, in this case RPE65, connected to a switch which turns it on, because without that the gene won't be expressed, and it's packaged in this shell, and so the virus is delivered to what's called the subretinal space. It's a space adjacent to the sick cells in the retina. It binds those cells, and then the DNA takes up shop in the nucleus. It starts producing the normal copy of the RP65 protein, and that corrects the biochemical deficit in those cells, which then allows them to function and allows the person to see.
That's incredible. we are talking about the viral vector, so I would also like to right now read out a couple of questions that have just come in regarding the use of the virus for the therapy. What happens to the copies of the bad genes that are being replaced in your eyes or bodies? That is one question that has just come in, Dr. Bennett.
Okay, so the bad genes stay there. In this particular disease, the bad genes are bad because they don't do anything. And so we don't need to remove them. They stay there, and instead these cells are gain the new functioning genes, which take care of the missing function.
Okay. There's another one. Does the virus used in the gene therapy treatment make you sick in any other way? Does it?
So I'll handle that
does
one.
It do anything else to the body? Yeah.
Yeah. So that the one of the most important things that has been seen in AAV research, and that's not just eyes. That's systemically large organs in the body where they're using amounts of this virus that are thousands of times more than what we're using in the eye. It's very, very safe. AAV, which is adeno-associated virus, is a recombinant or a genetically engineered parvovirus, which in humans does not cause any significant disease. There's some very esoteric condition called fifth disease, which most doctors have never seen, and it's really just a very mild viral illness. But that's the unmodified wild type adenovirus or adeno-associated virus. This has been basically any pathogenic or toxic factors have been spliced out of the virus, and that the virus is basically engineered so really all it does is it carries this piece of cargo, this new gene, into cells and does not produce any sort of toxins. So it is extraordinarily safe.
Thank you, Dr. Maguire. You already touched upon a little bit about the actual trial protocol. We're just trying to understand this a bit more. How much preparation should a patient or is a patient conducted through just before this injection or being treated with Luxturna?
Well, they're really, I mean, aside of the testing that's done before to make sure that they have the condition and they have enough viable cells to treat, they're really no different than any other person who's having a retina procedure in terms of the standard things: don't eat or drink after midnight or a few hours before the surgery, a general medical evaluation to make sure that there are no issues that might come up with the anesthesia, and really there's no significant difference, no major difference between any other type of eye procedure, eye surgery, and the Luxturna.
So how long does one procedure take? Like, for example, treating with Luxturna, what is the time for the surgery or the injection, that is?
So we know this well because we track times. So one way of putting it is in our original trial, it took about 2/3 the time it took to obtain an informed consent. The procedure takes about 50 minutes to perform from beginning to end. Certainly, the entire day, as I tell people, well, you have to show up to the hospital. We have to go through the checklist, and you have to stay afterwards for a few hours to make sure that you're properly drug can soak into the retina, but the actual surgery itself, about 50 minutes long.
And being an observer of all of these surgeries, I'd just like to add that whole time of surgery is mainly for preparing to do the injection, and the injection itself takes all of about 10 seconds.
Okay. Well, that's quick. So we also have Sarah Pettit with us. She's, as I just mentioned, she's Creed Pettit's mom. Creed has received Luxturna and after it was approved. So Sarah, can you hear me?
Yes, ma'am.
Hi, Sarah. Great to have you here. Thank you for joining us.
Thank you.
Sarah, so Creed has had Luxturna treatment for both his eyes, I understand. Were they administered together or one after another? Can you just give us a little bit detail about how the treatment went and how he's feeling now?
Sure. He had his right eye done on March 21st, and then we stayed down in Miami, and he had his left eye done on March 28th. And, I mean, he's doing great right now. He's very overwhelmed. It's a lot of things have it's like teaching a toddler at this point, a lot of new things that he's never done, but he loves it. He wouldn't, he's very happy with what he says all the time. It's his new eyes. Going out, like the doctors were saying earlier, how there was a curfew for the time, and that was so true. We were constantly fighting against the sun, and now there's no rush. We can hang out outside even when the sun is setting. He can read books outside now. He can play outside now. And he's much happier now that this is all done.
That's great to hear, Sarah. Dr. Bennett, this is a very common question I've seen on forums and people asking whether people can get treated for only one eye.
Theoretically, people can be treated with just one eye. In our experience, at least in the clinical trials, everybody wanted their second eye injected, particularly after the first eye was injected and they saw how much their vision improved. So, yes, I would say it would be possible to inject one eye, although the SPARC is recommending that both eyes be injected if they have enough cells that could merit the injection.
Yes, and Dr. Maguire, you mentioned a week of time gap between one eye injection and the other eye being injected. Is there a reason or some considerations that we take into account when we decide this time?
So there's a sort of a history behind the interval. So from a view, it's felt that if you give the exposure to the virus a very short period of time, you are not priming the immune system to the second injection, so it's not going to act like a vaccination. So theoretically, the immune system will look at two injections within a 2-week period of time as essentially being one injection, one exposure to this antigen, to this virus. It turns out, though, that one of the studies that we did earlier showed that patients who had the second eye treated in a safety study, the second eye treated sometimes years after the first eye, had no untoward reaction, had no major or minor immunologic response, and they also seem to have just about as much improvement in their retinal function as they did when we had a short injection interval. So that time period is really based on what was the minimum time between the two eyes, and just the idea that theoretically it might be better to have the second eye treated at a shorter interval to avoid an immune response.
Sarah, now that Creed is doing better, so what are some of the precautions that you take now that he's able to see and see better? That, as I said, improved, his vision is improved. So are there any precautions that you're taking other than, of course, as you just mentioned, you're teaching him like a toddler? So we'd just like for the parents who are listening. Yeah.
I take a lot less precautions than I did before. I have to say I unfortunately regret coddling Creed so much. I've watched for 9 years now other parents with children visually impaired all the way to blind, children who can ride bikes and button their clothes. And I was a single mom for most of Creed's, most of the time until recently. And so I kind of coddled Creed and protected him and did not want him to get injured, and made sure that he kind of lived in his own little bubble. And now that he had Luxturna, the one thing I do regret is not challenging him a little bit more before Luxturna, because it's definitely, to me, and to him, it's been very overwhelming for both of us, because he no longer wants me to be right there over his shoulder, and I'm having to learn how to step back. So my precautions, I think the only thing that I am concerned about at this point in time is poking himself in the eye or something silly like that. There's nothing anymore when it comes to him running into the table or tripping over the dog or just walking into people in general. Those are not my concerns anymore at all, and none of that actually happens anymore either. So it's like the tables have been turned on both of us. So I definitely have to learn to let go at this point.
I hope that Creed is able to see his rainbow very soon, because
yes,
I've been reading about him wanting to see a rainbow.
Yeah, and it has rained every day for months, and we still haven't had one. So I was like, this is ridiculous. So hopefully soon.
Thank you very much, Sarah, for sharing that.
Yes,
Dr. Maguire, how long does it take actually for patients to recover and able to see an improvement in vision?
So we have actually documented that the retinal function can improve in the matter of days. We've done special testing of pupil response, and basically as soon as we tested, I think around 5 days after the procedure, we were able to establish, in fact, there's much greater retinal sensitivity. It's interesting because there are certain tests you can't do immediately in the postoperative period, but you can certainly ask patients how they're, what they're seeing and what they think. And I was struck. I didn't really believe it at first, but now I'm pretty sure it's true. I was struck on the day after surgery, patients complaining about how bright it was, and it's almost an overnight effect where they'll get much more sensitive and seeing light's much easier for them. Fortunately, that almost overwhelming light sensitivity that they get patients get used to it after a few weeks, and it sort of calms down. So it's their new norm.
This is amazing, actually, to hear. And I can't but help smiling. If you could see me, I'm just smiling through this, hearing both of you talk about this great, absolutely wonderful treatment that now we have for the people who have LCA. So, Dr. Bennett, what is the data that we have on how long the patients retain their improved vision?
Well, we have more than a decade of data now, going back to the very first patients who were injected. The thing is, those first patients all received different doses, and so there are different variables that could affect their response. And all of them say that first injected eye is retaining vision, and we can measure that. However, our best data comes from the second eye that was injected, because all those second eyes got the same dose. And we are now nearing the 5-year time point on those patients, and so far they have maintained all of the gains that we measured early on, within 30 days after their injection.
Okay, so we do not have comprehensive numbers like 3 years, 4 years, or is that a progressive? Are you noticing a progressive vision betterment as they are progressing over the years? Is that how it is?
Well, the improvement basically it skyrockets early on. So by day 30,
okay.
People are seeing immensely better than they were before the intervention, and that what we're measuring there plateaus in terms of light sensitivity and other aspects of vision. However, we think that there are changes going on that we don't measure in a clinical trial, such as understanding what you're seeing. For example, we had one patient who was with us in a car looking out the window, and she had never seen tree branches before, and she had to be told what those were, and so she learned what tree branches were after her intervention. She learned what a reflection from a pond was. So these are a lot of abstract things that I think the person has to learn, and that's not something that we can measure.
Yeah, right. With that, actually, I think I should just now hand over to Tabby. She has some questions for you. Tabby, you're on air.
Hang on, let me unmute on my end. Can you hear me?
Yes.
Yep.
Okay.
Please ask your questions. Yeah.
Once you live in the blindness world and raising kids with low vision, I think we know what the term blindness is and how that's defined. We live in kind of this space where blindness and low vision is kind of like a spectrum. Can Dr. McGuire or Dr. Bennett, either one, put a little bit of definition, put a little bit of description around what it means to be legally blind?
So legally blind means two different things, essentially. There's visual acuity, which everybody is very familiar with. That's reading an eye chart. And there's 20/20 vision. I think of it as a fraction. 20 divided by 20 is 100% normal central vision, whereas 20 over 200, which is the threshold for legal blindness, that would be 10%. Basically 20 divided by 200, 10% of normal central vision, and that's the threshold you cross, and that's considered legally blind for center vision. What's interesting is there are numerous retinal conditions, forms of retinitis pigmentosa and inherited retinal diseases, where people have 20/20 vision and they're legally blind, because having good center vision is very important, but that represents only a tiny fraction, less than 4% of your entire visual field. So people are essentially with that sort of situation are looking through a long tube or a gun barrel. If your peripheral vision is constricted within 10 degrees of center, which is a little bit harder a concept to understand. It's not like visual acuity. It's a more abstract test. But if it's constricted within 10 degrees of the center, the normal being out to about 70 or 80 degrees, then you're so constricted that it impairs your ability to walk around and be mobile, to drive, to even do things like reading, and that's considered also a criteria for legal blindness. So there's visual acuity, which is 2200, and visual field, which is within 10 degrees, constricted down to 10 degrees.
Now, is it the blindness or lack of vision that qualifies people to come into the study? So I know there was some mention earlier about having functional retinas. So working with a lot of the parents and a lot of the kids that are in the LCA community, but also with other genetic disorders that affect vision, one of the big questions that we hear a lot is if kids or adults are already blind, can they still be treated?
So it depends on the mechanism of the condition, and I'll just comment on this RPE 65. So the cells, the nerve cells are still there, but they lack the molecule to capture light. So it's like having a car and with no gas in it, and it's. I wouldn't say it's completely unique, but it's a very unique situation. A lot of forms of blindness. It's not just the cells aren't working correctly, but they're actually degenerated. They're worn out and they're not there anymore. And that would require a different form of treatment than gene therapy, because there are no cells to treat. I think maybe Dr. Bennett could comment on other forms of how the biology works to cause the vision impairment.
Yeah, well, I would like to add that vision also depends upon the wiring being intact between the retina and the brain. And we don't know a huge amount about how long that wiring persists if somebody's retina is not functioning. There have been a number of very historic experiments studying this phenomena in newborn animals. But what we found with the RP65 was actually surprising, that even though the wiring between the retina and the brain hadn't been used for as long as 35, 40 years in one person, when we could treat the retina, we could activate that wiring and allow it to provide the information to the brain, which, of course, is what we use to have vision. So we don't know the answer for the other conditions, whether the wiring is still viable, but we certainly have optimism based on our findings with the RP65. And so, for example, if there is somebody with LCA who only has light perception but has never had formed vision, it's a big question. We think that actually the wiring would be intact since it would be primed by having the light perception. But could we then get that person to be able to see a face? We don't know, because we don't know if the brain is as malleable as we'd like it to be. But it's something that we feel that we've demonstrated in animal studies is possible. So we're hopeful that it will be effective for other genetic forms of early onset blindness.
One of the questions that comes up, or one of the conversations that takes place a lot within blind communities, is once they were identified as being blind or having light perception whenever they were younger or in their teen years, that they really don't have a lot of eye exams because they don't necessarily have a need to have them evaluated. They don't hurt, they just can't see. And so the question comes up of the discussion really kind of goes around the area of how do you determine if the retinas are non-functioning? I mean, we tend to think of it as visually, if you can't see, then they're not functioning. But what's the test that's actually done, and what's the advice that you guys give out to folks that may have an RPE condition, since that's the one we're talking about today, and how do they go about determining if they might be a candidate?
So one of the answers to that is if you don't get an exam, you'll never know. and you'll never have information like this. So if you had just, in the past, people just sort of threw up their hands and said, what am I doing this for? There's no treatments available, and I just get the same answer over and over again. And to be honest, I'm completely sympathetic to that because, I don't like to burden patients with unnecessary appointments. Everybody has other things they'd rather be doing. However, things have progressed. Things do progress, and over the last 10 to 15 years, for instance, we now have new methods that we combine with a lot of the sort of standard testing that allow us to make better determinations as to whether a person has the potential for vision. We can now actually image the retina with tomography at extremely fine detail, almost like a optical biopsy. So we can tell if the cells are there. They may not be functioning because the biochemistry isn't working. They don't have the proper molecules to capture light, but we know now we can do a very simple, painless, non-invasive test to measure the thickness of the retina or even to see the individual nerve cells. So it's a combination of things that we use to make the determination. It's not just photographs or measuring visual acuity or visual fields. We use a sort of consortium of things from our toolkit to be able to make that determination. And again, if you check in with the eye doctor periodically, you'll be able to take advantage of that kind of testing and the new technologies that come up.
Yeah, and just to add to that, we find now that we are trying to develop gene therapies for other forms of blinding disease that we're running double time trying to get patients with these diseases to come in so that we can adjust our protocol to measure the sort of responses that would be appropriate for those various conditions. Those are so-called natural history studies. And that's just because people have been told there's nothing you can do and have been lost to follow-up. And one more thing to add, maybe Dr. Maguire wants to comment on this, is for very young children, it's probably not a great idea to measure them too frequently because then they might get scared of going to the eye doctor. And I don't know if you want to comment on that.
Yeah, I think they need a diagnosis, but I'm very much an advocate of not creating phobias because I'm the one who has to take care of the people with the phobias that they develop during childhood. So if you slowly ease people into the examinations and have a good pediatric trained ophthalmologist get the initial diagnosis, that's great, and and then if they can be treated, great. If not, then just sort of check in every periodically to make sure that you're up on the new technology, and if there are any clinical trials that come up, they'll be made available to you.
While we're talking about young kids, one of the questions that we hear within the groups is, how do you guys evaluate? You talked a little bit about the acuity charts. How do you evaluate, and what eye charts do you use for the really young kids?
So I have been more and more impressed over time how sophisticated children are. So this study that led to Luxturna being approved included children from 3 years of age and older. And you would think 3 years of age, they had to do some pretty sophisticated stuff in terms of doing mobility courses at different light levels, and then being able to do vision field testing, et cetera, and by and large, most of the people who came for the study were able to do it and got included. So our youngest was near 3, and theoretically that's a good age to do the treatment. So, but to answer your question specifically, there are many different eye chart systems, some of which don't even require a verbal answer from a child. There's something called preferential looking, that a masked observer will look to see if a child looks at a certain side of a chart more than the other, and they can tell the preference is usually gives an indication if they're able to see something. You can also ask a child to have their own card with the letters or figures on it, and they actually will point to them on a card so that they don't have to give a verbal answer. And the very simple thing is a HOTV chart, which use just those 4 letters, what we also call letters optotypes, but only those 4 letters. And most children, if they can't read the entire alphabet, can certainly be taught those 4 letters, and that is a early sort of verbal chart that we use. But the kids are smart, are a lot smarter than they sometimes let on.
Now, are you guys, over the course of time, I think we're talking about 25 years, are you seeing that the research is indicating that the treatment impact is potentially more robust when the treatment is administered in young populations, as young as possible?
Gene?
So our initial results in the animal models definitely showed that there was a much better response in those younger animals than the older ones. And it makes sense because this is also a degenerative condition. So over time, the cells die off and there are less of them left to treat and thus to respond. And so we hypothesized we would see the same thing in humans. And certainly the results of the intervention in individuals of a wide range of ages in humans show that generally the outcomes are people perform at a much higher visual level the younger they are, probably reflecting the number of cells that they have, and this is sort of a generality. However, it will take us more time to analyze all that data in an objective fashion to clearly get a readout. But certainly, one would argue that it would be more advantageous to a person to have vision over a longer span of their life than if they were treated when they were older.
So the burning question for a lot of folks out there that might have low vision or have no vision at this point in their life and have kind of fallen off and not necessarily been to an eye doctor recently, how exactly do you determine if they have the RPE mutated genes that qualify or that has the treatment available now?
It's really quite standard now for people to provide a saliva sample or a blood sample or any other tissue, a skin sample. Typically, it's easiest to give a saliva or a blood sample. And then DNA is isolated from that sample and then screened for presence of mutations in a panel of genes. And sometimes that panel is limited to those that cause early onset blindness, but there are now more than 265 different genes known that when they're mutated will cause blindness. So the testing has gotten really all inclusive in most cases, screening for a large number of potential gene mutations, which provides even more robust confidence that when you find a given mutation, that is the one. And then typically one confirms that is indeed the disease-causing mutation by testing the parents and seeing if each one of them is a carrier of one of the mutant copies that the child has. And that adds further confirmation to the genetic diagnosis. There are now. Go ahead. I'm sorry.
Go ahead.
There are now many testing laboratories, not only in this country but around the world, and much faster feedback from the test results.
So representing a lot of the parents that are in the LCA group and the kids that are coming up through it as well, we're all pretty well versed and aware that it was the dogs that initiated a lot of this work that has moved through to a clinical trial and now moved into a treatment. So one of the questions that I hear among the kids in our group is, how does the surgery that you're doing on your patients, your human patients that come in today, even though it's an FDA-approved treatment, how does that compare with the surgery method and all of the things that you did with the dogs whenever you were in the research phase?
So the drug is a little bit different because it was modified. It was humanized. Is that the term we use?
Sure. The human
gene,
cDNA.
Yeah. So dog's DNA is very, very close. The gene is almost exactly the same, but not quite exactly. So when we were testing the original dogs, it was more canine-based in terms of the gene that was introduced. So the surgery is essentially the same. You take a small tube, place it under the retina, and inject the medicine. The actual mechanics are a lot different because imagine, dogs have their eyes on the side of their head. They have this big snout in front of them, which would, if you were in a human operating theater, that would hit the microscope. That would be a bit of a problem. So a lot of it was just little sort of mechanical things to properly position the animals and just compensate for different morphology, different look to the face and to the snout. But when it came down to the actual treatment, they had a injection under their retinas exactly like the humans, and they respond in an amazingly similar way too.
Yeah, I'd like to add that two of those initially treated dogs are now living at our house. At the end of the experiment, when we had all of our data, we decided they had given us such great information and been so valuable, and we loved them in addition, so we actually went all the way to the provost at the university to be able to adopt them and give them a good home.
You guys, can you share with everybody out there, especially the kids that are listening, what are the dogs' names? And what type of dogs are they?
So it's Mercury, Mercury and Venus. Venus is the mother. Mercury is the son. And they are Swedish Briards that were bred by Christina Narstrom, who's a Swedish veterinarian. And they are very unusual. You can Google image Swedish Briards. In fact, I think their pictures are probably going to come up. But they're very unusual and very beautiful looking dogs. And they run around the backyard and they look, bark, and move around just like any other dog I've ever had.
And they're very, very sweet. If any child listening would like to meet them, they're always welcome to meet them in Philadelphia.
Now, a curious question, just as dog owners out there. Once they received the treatment, you had mentioned that they had the same type of rapid pickup. Did you find that there were odd little things that you kind of had to reteach them, as we were talking about with Creed and some of the other things that we're seeing go on?
Yes. So these animals were born and raised in an animal facility, which is on one floor, and there are no windows, no grass. So the first time that I got approval to take the dogs home, I brought them outside, they saw grass and they're pawing it because they'd never seen anything with that texture or shape, color before. And then stairs were a new phenomenon for them. They'd never seen stairs, and they had to kind of figure out, oh, this is how you walk downstairs. Now you'd never know there was a problem, but that they had never had that experience, and it's not a problem for them because they can see the stairs, but it was a new experience.
That's wonderful. Thank you for sharing that.
Thank you, Tabby. That was a great set of questions. Dr. Maguire and Dr. Bennett will take some questions from the audience now. We have a whole lot of questions on the website. I see the most common one is if we see that the vision is reducing again, do we do this procedure once more?
So we don't have an answer for that, just because I guess it's a good problem. We haven't definitively seen in the phase 3 the trial any reduction in vision. And I guess part of the thing is, why is the vision being reduced? So when you create one of these blebs, when you inject under the retina, we're not actually injecting under the entire retina. That would require, that would be more of a risk, and it would require a lot more material. We try and treat the most relevant parts of the retina for day-to-day vision, the center vision, and things you used to walk with. So it may be that the areas that don't receive the drug aren't working quite as well, and you might be able to measure that as a slight decrease over time because those areas aren't receiving the drug. But I don't know that that would be a good reason to go back and inject, since those parts of the retina really aren't used very much for vision, though we can still measure. But yeah, it depends what the reason the decrease in vision and why it happened. It's conceivable that we might re-inject or do additional injections if it was the case that vision were decreasing and we could improve things by additional injections.
Thank you, Dr. Maguire. We have some questions regarding the age or the range of age that patients are selected for the treatment of Luxturna. Also asking if newborn screening is possible, and also if adults can be treated.
So, Jean, why don't you take the newborn screening, and I'll talk about the age range.
Sure. Okay, so for newborn screening, yes, indeed, it is possible to do that. At present, newborn screening is not covered by insurance. However, it's possible to do that on your own, and that could, again, be done from a tiny blood sample or saliva sample, or if skin is removed for some reason, that can be tested. However, I would like to add that if the individual is found to have an RP65 mutation, the current guidelines for delivery of Luxturna require that somebody be 1 year old or older, so that there is a large time window during which this testing could occur prior to a treatment.
So in terms of the age range, as Jean said, it's 1 year and older, which is it was thought that at that point the eye is large enough that the treatment could be administered with minimal increase in risk. And in addition, the nerve cells in the retina are done replicating, so there would be no dilution of the drug, into more and more cells as they divide. In terms of the oldest, it all depends. There are people that we've seen who are in their third, fourth decade of life who actually have a population of cells that are still there and can potentially respond. So again, it depends on. There is no upper age limit as long as there are viable cells that are left and can be demonstrated. Unfortunately, there are some people we've seen who are in their fourth, fifth decade of life, and honestly, they just don't have any population of nerve cells left, and for them to go through a procedure like this would just be it doesn't make sense, and it would actually be no advantage to them at all.
Thank you, Dr. Maguire. So another question, I think quite a few questions on whether this kind of approach can be used to intervene in blindness conditions which are due to other mutations, and especially a research on CEP290.
So there is a great deal of laboratory research that is ongoing all over the world, looking at ways of intervening with other forms of retinal degeneration, including CEP290. The particular challenge with CEP290 is that the gene is very large, and so it does not fit within the cargo capacity of the adeno-associated virus, the AAV, that we spoke about. But there are some other strategies that are being considered, and including strategies of allowing this gene to fit into that small cargo capacity. So back to the original question, the answer is yes. If the gene can fit and it can be delivered into through the AAV to the appropriate retinal cells, it should work to intervene with the condition and provide vision. And indeed, proof of concept has occurred in multiple genes in animal models in the laboratory, and is forming a pipeline that hopefully will translate into human clinical trials in the near future.
Thank you, Dr. Bennett. I would like to read out a comment that has just been posted to both of you. It says, "I'm so grateful for this groundbreaking technology and the people who have made this possible. I have a 10-year-old daughter who is at her 2-week post-op in one eye and 1-week post-op on her other eye today. We have already noticed big gains in dim light. She is seeing things in a whole new light and is so excited about her new perspective. As a mom, to see her point to the sky and say, 'I see clouds,' nothing is more beautiful. So thank you, thank you, thank you." That's what she writes. So Dr. Bennett and Dr. Maguire, I think you've made many, many children happy, their parents happy, and 25 years of research on congenital blindness. That's awesome. Has finally brought in this approval of Luxturna. And thank you so much. We're almost at the end of our hour. Thank you so very much for your continuous, persistent efforts, your time, and sharing all this information with us. We'll also like to thank Tabby and Sarah for participating, and Tabby for those excellent questions for your group. And also thank University of Pennsylvania and Children's Hospital of Philadelphia and the audience. This talk is recorded and will be available on curetalks.com. So please visit our website for details on more upcoming talks. Thank you and have a great evening, everyone. Thank you.
Thank you.
Thank you so much.
Thank you.